Okay, hello, good morning, everyone. Hello, today I will present a study titled Isolation of Microplastic in Biota-rich Seawater Samples and Marine Organisms. It's written by Matthew Coles and wait. 好，然后 okay, so next, let's go to the page 1. I will slightly introduce the microplastic. So with the widespread use of the plastic product, microplastic pollution has become a significant global environmental issue, particularly affecting marine ecosystems like a range of aquaculture marine species will eat into the microplastics. Microplastic litters has been identified in marine habitat across the globe and more recently has been sampled in freshwater systems. And those aquatic organisms, including zooplanktons, benthic invertebrates, bivalves, fish, and seabirds, ingest plastics, which may result in reduced feeding, energetic deficiencies, injury, or death. And the microplastics separate like three kind of it. The first one is the primary microplastics. It's manufactured as a small plastic particles such as microbead in the face wash and in industrial materials. And the second one is the secondary microplastics. It's breakdown from the large piece, like 保丽龙或是pet, okay. It's breakdown from cause the sunlight of wave from the plastic bags or fish net. And then third one is the synthetic fibers. It's released from our clothes or other made by fibers while we washing it, or when it touch the waters, it will through the wastewater to the ocean. And the second one, we will introduce the microplastic. It's cause it's 疏水，它 cause the plastic 表面的疏水性。It may contain harmful chemicals such as facilities and bisphenol A and PAH. It's another environmental hormones. And this toxic substance can accumulate in marine organisms and be transferred up to the food chains, ultimately affecting high-level predators, including humans. And next, we will talk about our research objective. The scientists need to find a new way to avoid dissolve the plastic litters when they're trying to clean the sample's biological organisms. So the paper will compare the three kinds of digestion ways to show this enzymatic digestion's benefit and advantages. Let's come to the methods. The first one is the sampling and preparation. The step one is the zooplankton sampling. They choose the station just below the England island. You can see this way, the Plymouth. The laboratory is in Plymouth. Okay. They use subsurface tools with 200 to 500 micrometers net to trap the zooplanktons. And these samples will send to the lab in Plymouth in 2 hours. And the sample preparation is a step 2. They will flush with purified water to desalt. And the purified water is made by Molecule, and they will use the oven to dry it at 6 degrees for more than 24 hours. And the third step, it's through the whole experiment. It's a mitigating contamination. They will all apparatus will wash and rinse thoroughly with Molecule, and the sample and the equipment were covered wherever possible to minimize period and exposure. Procedure blank, and they will leave a part of sample to through the microscope and make sure they will not contain the pollution plastic from the lab. Okay. And to the digestion way, the step 4 is they will use in the acid in HCl, hydrochloric acid, and the alkaline use sodium hydroxide to treatment the sample. And using the third way is the enzymatic digestion. They also use the sodium hydroxide with the proteinase K to digestion the sample. And after the digestion, they will put the sample to the ultrasonication. And using the HCl and the enzymatic digestion, they will directly put the samples on the ultrasonication with ice to assess its overheat. And after the digestion and ultrasonication, they will put to the vacuum filter and only retain the clear the biological material using pure water to flush it out. And the filters remove and put these samples back to the oven to dry it at 6 degrees. And they put 5 types of microplastic to the samples to compare the 3 ways digestion efficiency and efficacy. And you can see this is 5 kind of microplastic we will see in the next page. Next page. So this is a result about its efficacies. You can see the least effective treatment was the hydrochloric acid. It's only 82%. And comparative, the NaOH digestion is sampled by 90% and 91%. And the original enzymatic protocol is result by only nearly 90%. But the optimize is also the scientists just change the enzymatic protocol to using another way, like more time, higher degrees. They will let the digest more than 97%. It's significantly more effective than the other treatments using enzymatic protocols. And after ultrasonications, we just take off the HCl1. You can compare the NaOH and the proteinase K. You can see the NaOH digest sample is only 90% and then 87%. But you also can see the newest protocol can stay on the 97%. It's more, and the Protonix case less effective and damage the plastic samples. You can see the first one is the original microplastic sample, and the second row is after the enzymatic digestion. It's nearly slightly different, but after the alkaline digestion, it's significant different because you can see the nylon fiber, it's just disappear, and the polyethylene is just get like a okay, and the polyester is screwed like is, and they also use the fluorescent polystyrene microplastic to internalize by the pigment colloid. You can see the fluorescent microbit, and they use the same way in enzymatic to mineral the sample, and you can see the fluorescent microbit is stay complete. Okay, so discussion. Why is the enzymatic digestion is better? The traditional method, like these two kind is damage the pH sensitive plastic, like nylon.
What does the breakthrough in this area, in this study? Procedure break, what the is at in this study?
Blanks all. Okay, so they just want to make sure they will not get the pollution by the human, and they just want to make sure they will not get the pollution by the microplastic, because if we wear the clothes made by the plastic, they will also contamination these samples.
I want to know, this paper have two methods to sample the plankton?
Three.
Three? Okay.
They got 3 ways to digestion the plankton sample.
Okay, so I want to know which one is better or is useful for sample.
In my conclusion, the enzymatic digestion is significantly better than the other two kinds, like acid and alkaline digestion, because it's highly efficiency than the other two kinds, and it's more detect microplastics ingested by the zooplanktons, and it can quantify microplastic concentration in seawater. Okay.
Can you talk again about this different about it? Because I can see very well.
Okay, so the first row, this one is original shape, and the second one is after the enzymatic digestion. It's slightly different, right? And the third row is after the alkaline digestion. You can see it's significantly different after the digestion in alkaline.
Okay, thank you.
So it's damaged the sample.
Why you use two kind of size?
Oh, so there's a typo. It's only 200 to 500.
Between.
Between.
Thank you.
Sorry.
You probably know that you were reading your script very often and you were really looking like this. I hope that next time you will be more familiar with your script. And another issue is you need to speak louder, because when you speak lightly, it will give people the impression that you are not confident. That's how light speaking, the impression that you will give to other people that you are not confident with what you are saying. So you really need to speak louder. At the very first here, before you introduce these three, you had a very long line, super long, which is not related to any of the text here. So I was wondering which one, you have three here, which one were you talking about? But the fact was you were not talking about any of this. So when you have long line, please produce another slide describing that will fit your line better. For example, I think what was missing in your introduction is you talk about the introduction of microplastic and its pollution. However, you probably, here you have, here you introduce different kind of contaminants, and here you talk about marine organism. I think the marine organism deserve one specific slide because that's the main purpose of this study, because I have heard a lot of paper about microplastic every year. And so the study of microplastic, the method is not new. However, this one is new because this is specifically for zooplankton, right? So you need to say that clearly, that this is specifically for not free microplastic floating around in seawater, but microplastic ingested by organism. So it's a method for that. You need to say that clearly. Whatever, but they use zooplankton as example, right? Yes. So because for free microplastic, you don't need this complicated issue. I mean, microplastic in seawater. So please provide another slide to describe the background of the study better. And of course, you need to figure out what is the English of 保丽龙. Okay, how? Here you have an objective, but I don't think you actually introduce this. I would prefer that you put this in your material and method where you talk about, this is the zooplankton collected by the trawl, right? It would suit the material and method better. And I don't know where is this place. You need a regional map showing us where is this place. The key issue why you couldn't finish your talk in 12 minutes is because you have too detailed methods, far too detailed. To me, this can be condensed in 2 slides. One is 1 slide showing your sample and maybe the photo of your zooplankton sample, and you don't need this detail. And the second is this one, and I would prefer that you would use image. Okay? Because when you have these wordy slides, it's really uninteresting. I really want this. And you need to probably explain, because not all of them they have studied biochemistry. Not all of them know what is proteinase K. You don't need to show very detailed. You only need to tell us what does it do. What does this enzyme do? So too much detail. Yeah, you have how many? 1, 2, 3, 4, 5. Okay, that's too much. You only need 2. And use image. You can put ultrasonication and this in one slide, right? When you use image, we don't need these details. We only need image. Okay, that's all we need. And when you were introducing your results, you did not introduce what are these, what is that. For example, how did you define digestion efficiency? How is that introduced? So to me, these can all be condensed and maybe put here, below here. And all you need to, you can put, when you can remove these detail here, you can make the figure larger, and all you need is a concluding remark, 1 sentence, which is, which one has higher efficiency? That's all you need. Otherwise, Adam won't need to ask that question, right? Because you didn't make to the conclusion, right? And the same here. Bring the detail down here, enlarge the figure, and give 1 simple concluding remark. And here, you didn't do it well during your talk, but when you answer Jason's question, or Kelvin's question, it was better. Like you introduce each of them carefully. But you didn't do that during your talk. You were like, because you were almost running out of time. So I hope that next time you can take time, because this kind of figure is more like eye catcher. It's more interesting. So I hope that next time you will have time to introduce them carefully, and maybe you can print some keyword like this is damaged, this is dissolved. Print some keywords and not this kind of long sentence. Okay. Print keywords directly on the photo. Yes, use keywords. Okay. And here, this is also eye catcher. Okay. So I would avoid this long sentence, and you only print keywords on the photo. For example, this is microplastic. You just say microplastic. And this is after removing this animal, right? The remaining microplastic from this animal, right? So you can maybe put this figure here and then tell us what has been done in between, and that's the remaining microplastic, right? Okay, so I hope that next time you can make it here. Okay, so thank you, Sean.